Hi guys, hope you are doing well and you enjoyed those secret messages. I hope they were life-changing for you. This video is going to cover two topics, post-transcriptional modification and then go more into detail on translation. So to go back and review a little bit what happened on the last one. So in protein synthesis, we have two steps essentially. We have transcription, when the DNA is copied into an mRNA, and then we have translation, where the mRNA is read by the ribosome and translated into an amino acid chain, which will then fold into a protein. Well, there is actually like a little step in between these two steps, and that is called post-transcriptional modification. So after the mRNA gets copied off of the DNA, there's a little bit of a modification that has to occur to it before it's ready to go out to the ribosome. So at this point, it's actually known as pre-mRNA when it's freshly copied. One thing that has to happen to it is called splicing. In splicing, certain sections of that mRNA are cut out, and then the remaining sections are put back together to make the molecule. A lot of the DNA is not actually coding for anything. Sometimes we call it like junk DNA or junk mRNA or filler or something like that. It turns out that some of it probably does play a role in some other processes, but just for this protein synthesis part, these are bits that we don't actually want in the final protein and the final mRNA. So the quote-unquote junk bits, which are called introns, get cut out, and then the usable bits, the parts that we're actually going to make the mRNA out of, which are called exons, are glued back together, and that is splicing. A note, this is something that only occurs in eukaryotic cells, so prokaryotes like bacteria do not have this process. One of the advantages of splicing is that there are some mRNAs or pre-mRNAs that can be spliced in multiple different ways to actually make different genes and make different proteins. So for example, if this pre-mRNA up here is spliced this way, then it can make this protein, and if it's spliced this way, it can make that protein. So for not every gene, but for some genes, there are sort of multiple paths where you get two different proteins out of one section of DNA, essentially. The other parts of the post-transcriptional modification are known as the cap and the tail. So on the 5' end of the mRNA, a special guanine is added, and we call that the cap. It just kind of signals the cell that this isn't something that should be like broken down or destroyed. And then on the 3' end, just a whole bunch of adenines are added onto there. So it's just like 50 to 250 adenines, which we call this a poly-A tail. And that's similarly to protect it from degradation by the cell. So once those 3 steps are completed, the splicing, the guanine cap, and the poly-A tail, then the pre-mRNA is ready to finally be a full mRNA, exit the nucleus, and head out to the ribosome for translation. Now to quickly review our translation process. In translation, the ribosome clamps on to the mRNA, and then the tRNAs, shown here, are going to bring over the amino acids according to the codon on the mRNA. So if we look a little more closely at this tRNA, they each have a specific amino acid on top, so one of the 22 on top, And then on the bottom, they have 3 bases called an anticodon. So the anticodon matches up with the appropriate codon on the mRNA transcript. It's that beautiful system of base pairing that's carried us through all of this DNA and protein synthesis stuff. So for example, the tRNA that has CGG as an anticodon will pair with the mRNA that has GCC as its codon, and that tRNA will hold a particular amino acid up here that goes with the codon GCC. Looks like that's going to be alanine up here. One thing to remember is that the chart you have for which codon matches up with which amino acid, that always refers to the mRNA. The codons are on the mRNA, so not the DNA, not the anticodon. The chart is for the mRNA. So now let's look at the ribosome a little more closely. The ribosome is made of 2 parts, 2 subunits. There's the large subunit up here, and then the small subunit down here. And what happens with these 2 subunits is they come together and sort of clamp around the mRNA right here. You can think of it as like 2 hamburger buns, and then this is the hamburger, I guess, in the middle. You close it around the middle. Within the large subunit of the ribosome, there are 3 sort of slots where tRNAs can go, 3 places that tRNAs can kind of plug in. We call those the A site, in the middle is the P site, and over here is the E site. This thing right here is representing a tRNA, and then here is its amino acid, there's the anticodon down there, and there's the codon. Okay, so I'm going to explain this using the pictures. However, this is really a lot easier to understand if you see it in motion, like in a model in a video. So I would highly recommend that after this you check out some of those videos linked in the extra resources folder, because I think it's just going to be easier for you to get a feel for what's happening here when you see it in motion. But here we have, again, the gray is our ribosome, here's our mRNA down here, here's our A, P, and E sites, and then these guys are the tRNAs with their anticodons down there, and then these are our amino acids, which are going to be forming that chain to make the protein. So, first thing that needs to happen is a tRNA needs to come in to the A site, and then once that tRNA is at the A site, if this is the first one, it's actually going to slide right over to the P site. Then another tRNA is going to come into the A site, and the amino acids that are attached to these tRNAs, the one at the A site and the one at the P site, are going to be bonded. Okay, so they're going to form a bond, because remember, we're trying to link amino acids together to make a chain to make a protein. Then everything is going to slide down one slot. So the tRNA that was at the P site moves to the E site, the one at the A site moves to the P site, and then a new one slides in to the A site. The one that was at the E site, as it moves to the E site, it detaches from its amino acid, and then it leaves. Go pick up another amino acid. And then the new one that's at the P site is now going to be holding the growing chain. Its amino acid gets bonded with the new one at the A site, and then everything slides down again. So everything slides down one codon each time, 3 bases, and it's this process that continues until we build the protein, until we get to that stop codon, at which point the protein is completed, everything detaches, and then the amino acid chain leaves and goes away to go fold up. All right, so that's it. That's the process of translation. Again, I would recommend watching a video or two to see this more in action. And please let me know if you have questions in the discussion for this lesson.